Hello everyone, welcome back to my YouTube channel. Myself Professor Shahana Sengupta. Presently I am associated with Swami Vivekananda Institute of Modern Science, Kolkata as an assistant professor in the Department of Microbiology. In my YouTube channel, I generally share different types of academic videos related to the field of biological sciences. In my previous video lecture, I have discussed about the monoclonal antibody and its production procedure. In today's video, I would like to discuss about different types of monoclonal antibodies which are having enormous importance in the field of clinical immunology. Now, I would like to discuss about different types of monoclonal antibodies. The four major types of monoclonal antibodies are MURINE monoclonal antibodies, CHIMERIC monoclonal antibodies, humanized monoclonal antibody, and human monoclonal antibodies. All of them are having important therapeutic application. Productions of monoclonal antibody was started with murine antibody productions by means of hybridoma technology. Later, due course of time, other types are discovered and modified. Let's begin with murine monoclonal antibodies. Murine monoclonal antibodies were the first antibodies ever to be produced at lab scale by hybridoma technology in the year of 1975. They are known as first generation monoclonal antibody. The procedure of monoclonal antibody production through hybridoma technology would yield monoclonal antibodies whose 100% amino acids are mouse derived. The problem of mouse monoclonal antibody is that the mouse protein being foreign can induce immune response in humans, producing human anti-mouse antibodies or HAMA, which in turn eliminate the monoclonal antibodies faster from the body. Hence, mouse-derived monoclonal antibodies are not the best for human use. Since the discovery of hybridoma techniques, various modifications have been attempted to produce monoclonal antibodies by recombining human and mouse proteins. To overcome the problem of murine monoclonal antibody, there occurred the introductions of second generation monoclonal antibodies. The second generation monoclonal antibody productions are completely based on DNA technology or genetic engineering. With the help of those technology, there occurred the construction of this type of antibodies by using the hybrid composed of human antibodies regions with murine. This type of second generation monoclonal antibodies were much more effective and free from previous problems. The examples of second generation antibodies are chimeric antibody, humanized antibody, and human monoclonal antibodies. Let's discuss about chimeric monoclonal antibodies. Chimeric monoclonal antibodies are structural chimeras made by fusing variable regions from one species like mouse with the constant region from another species such as human being. This is an ideal fusion. Antibodies are approximately 65% of human made. This reduces immunogenicity and thus increase the serum half-life. After insertion of human and murine genes into a plasmid and transfection into bacteria, chimeric antibodies are produced as inclusion bodies. Following lysis of the cells, the chimeric antibodies are purified for in vivo use. Many therapeutic monoclonal antibodies were originally generated in mice and then chimerized to reduce immunogenicity in humans before humanization was widely adopted. Examples of chimeric antibodies in the clinical sectors includes inflixi monoclonal antibodies, rituxi monoclonal antibodies, and AB6C monoclonal antibodies. Let's focus on humanized antibodies. Humanized antibodies are antibodies from non-human species. This type of antibodies made in laboratory by combining human antibody with a small part of mouse or rat monoclonal antibody. The murine hypervariable amino acid domains are grafted into human antibodies. This results in a molecule of approximately 95% of human origin. The mouse or rat part of the antibody binds to the target antigen, and then human part makes it less likely to be destroyed by the body's immune system. This binds weakly to the antigen. A humanized antibody has three major advantages over the original mouse monoclonal antibodies. Like the immunogenicity of the monoclonal antibody is reduced since much of the immune response occurs against the mouse immunoglobulin constant region. Secondly, the human constant region allows for human effector functions to take place. And lastly, the serum half-life of the monoclonal antibody in human is increased. Let's discuss about human monoclonal antibodies. Human monoclonal antibodies are produced by transferring human immunoglobulin genes into the murine genome, after which the transgenic mouse is vaccinated against the desired antigen, leading to the production of monoclonal antibodies. A simpler method of constructing human monoclonal antibodies can be accomplished by using recombinant DNA techniques. Once a mouse monoclonal antibody has been constructed using the traditional method of hybridoma technology, DNA encoding the antigen binding portion of the antibody molecule can be isolated and fused to human DNA that encode an antibody. Then the hybrid DNA is inserted into a bacterium which produces half mouse, half human monoclonal antibodies. The antibodies made by this method are less likely to induce an anti-antibody response when given to humans. Further fine-tuning can be done to change all parts of the antibody that are not directly involved in binding to the specific antigen. This technique has been used to produce large number of different monoclonal antibodies for use in therapy. It contains 100% human-derived amino acid. It is the best accepted monoclonal antibodies in human. However, it is difficult to produce human monoclonal antibodies by conventional hybridoma technology. There are also some limitations that is associated with these human monoclonal antibodies. For ethical issues, humans cannot be immunized against antigen, number one. Number two, the fused human lymphocyte mouse myeloma cells are very unstable. Thirdly, there are no suitable myeloma cells in human that can replace mouse myeloma cells. I would like to conclude my lectures after discussing some other types of monoclonal antibodies which are having important clinical applications. Let's begin with naked monoclonal antibodies. There is no drug or radioactive material attached to them. They work by themselves. These are the most common type of monoclonal antibodies used to treat cancer. Most naked monoclonal antibodies attach to antigen on cancer cells, but some work by binding to antigens on other non-cancerous cells or even free-floating proteins too. Naked antibodies can work in different ways. Secondly, conjugated monoclonal antibodies. Monoclonal antibodies joined to a chemotherapy drug or to a radioactive particle are called conjugated monoclonal antibodies. These monoclonal antibodies are used as a homing device to take one of these substances directly to the cancerous cells. The monoclonal antibody circulates throughout the body until it can find and hook onto the target antigen. It then delivers the toxic substance where it is needed most. This lessens the damage to normal cells on other parts of the body. Conjugated monoclonal antibodies are also sometimes referred to as tagged, label, or loaded antibodies. The third one is bispecific monoclonal antibodies. These drugs are made up of parts of two different monoclonal antibodies and they can attach to different proteins at the same time. An example, blinatumomab monoclonal antibody, which is used to treat some types of leukemia. One part of blinatumomab monoclonal antibodies attaches to the CD19 protein, which is found on some leukemia and lymphoma cells. Another part attached to CD3, a protein found on immune cells called T cells. By binding to both of these proteins, this drug brings the cancer cells and immune cells together, which is thought to cause the immune cell or the immune system to attack the cancer cells. Thanks a lot for your patience. Please do subscribe my channel. Stay tuned for the upcoming videos in different fields of biosciences.